Welcome, everyone. Thank you all so much for being here today for this year's David J. Ramsay Entrepreneur of the Year Award. We have a wonderful hybrid program prepared for you all today. My name is Jenna Frick. I'm a Senior Media Relations Specialist for the Office of Communications and Public Affairs, and I'll be moderating this event today. So since this is a hybrid event, we have an in-person audience as well as a virtual audience who I'm talking to on the screen right now. So just a couple of housekeeping things to get out of the way before we jump into the program. For all of us who are joining us in person here today, when we have a question asked, make sure that you speak into the microphone. We'll have you just raise your hand during the Q&A portion after the main speaker presentation. And then we want you to make sure you speak into the microphone so that our virtual audience can hear your comments and questions as well. And for our virtual audience joining us over Zoom, if you have a question to ask during our Q&A portion, just go ahead and type that into the chat window of the Zoom, and I will go ahead and relay that question to our speaker. And that will happen after our main presentation. Also, our speakers today, once you come up, you make sure you speak into the microphone so that our virtual audience can hear you. And with that, I would like to turn things over to UMB's president, Dr. Bruce Gerald, to introduce our winner.
Greetings and thank you, Jenna. It's a pleasure to be here and to welcome this group. It's nice to have another hybrid program. So I hope things work out well and we get used to this because I think it's here to stay for a while. So good afternoon. This is the second event in the 2021 UMB Founders Week. We had our gala Saturday night where Vincent also received his award. Dr. Njar, it was a pleasure to see you there and all of your family.
Thank you.
There was quite an entourage and it was wonderful to see that group. So thank you for being this. We renamed this program a year or so ago when we had the sad loss of Dr. David J. Ramsay. Dr. Ramsay was president of this institution for quite a number of years and was really the believer and the founder of the biopark, really made that happen. That was an empty piece of ground and his efforts and his vision made that happen. And what a great legacy that is to have left here for us at UMB. We do celebrate entrepreneurism and innovation at this university. And it's always remarkable the different individuals who speak at this event. And I can remember many of them and their riveting discussions, and then followed by Phil Robolato talking about the many patents that this institution gets as well. So it's a great event. This particular award is made to someone who's made tangible efforts to improve the human condition, to come up with an invention or a discovery or some type of innovation that has the potential to help people and also to be developed commercially. It is evaluated extensively, and we pick from a large group of people to finally end up with the winner this year being Dr. Vincent Njar. I have to tell you, I know Dr. Njar for a long time. It's been a long time, hasn't it, Vincent?
Yes.
It's just been at least 15 years, maybe longer.
I think it's longer.
Yes, okay.
Almost 20 years, Bruce.
Somewhere here, and he's been here a long time, as have I, and he's contributed to this institution in many, many different ways. And so it's certainly a pleasure to be able to be here at this event for him. So I want to thank you all. With that, I'd like to introduce Dean Albert Reese, Dean of the School of Medicine, Executive Vice President for Medical Affairs, who will introduce our speaker more formally. Dean Reese.
Thank you, Dr. Gerald. It is a pleasure and a privilege to be able to introduce our speaker this afternoon. Before I do it, I also, as Dr. Gerald did, want to say a word or two about Dr. J. David Ramsay, who I was fortunate to have been hired by Dr. Ramsay in 2006. It just so happened that yesterday afternoon, I received a book that is called Remembering David J. Ramsay. And it's written, it's really prepared as a collection of remembrances prepared by many of his colleagues, his friends, students from his time in the United States. I can only say, David, as the book very nicely puts, was a wonderful, gentle, caring, thoughtful, kind person who spent 16 years as president here at the university, and for which we are most grateful in launching this and other things. So very nice way to honor Dr. Ramsay's memories and his efforts. Well, in that vein, Dr. Vincent Njar is he the inaugural Dr. Gerald? The inaugural David Ramsay? Second one? Okay. Well, you are the second, but that's a very important distinction. The second David Ramsay Entrepreneur of the Year. So let me give you a brief summary of Dr. Njar's work here and and the work he has been doing. Dr. Njar is a professor in the Department of Pharmacology, and he's head of the section of medicinal chemistry in the School of Medicine's Center for Biomolecular Therapeutics. Dr. Njar is an entrepreneur par excellence. First and foremost, he's an outstanding scientist. He has more than $2.5 million of extramural research funding active at the present time. Dr. Njar has had a long-standing interest in the discovery and development of small molecules as anti-cancer agents. His discoveries have led to the development of novel therapeutics for breast cancer, prostate cancer, and pancreatic cancer. These are just some of the most prevalent and deadly cancers in the world. But most importantly, these are the types of cancers that Dr. Njar's work is aiming toward impacting directly. In fact, his collaborative work with the late Dr. Angela Brody resulted in the discovery of aromatase inhibitors, which at the present time is now the worldwide standard of care for treatment of postmenopausal estrogen-positive breast cancers. Dr. Njar's deep investment and his interest in has made him a world-renowned scientist. But entrepreneurs are also doers. They turn interests and ideas into actions and into tangible products. Through his skill and will, Dr. Njar has commercialized his discoveries. Over the past 4 years, he has founded 2 companies to direct the early development of his inventions and technologies. In 2018, Dr. Njar partnered with UMB's New Ventures Initiative to found an early-stage small molecule oncology company to develop early therapeutics for triple-negative breast cancers and other cancers as well. For his profound and creative research and his collaborative and congenial style of finding solutions, Dr. Njar is what I would describe as the epitome of what a biomedical entrepreneur should be. Dr. Njar, it gives me immense pleasure and pride, in fact, to introduce you as the 2021 David J. Ramsay Entrepreneur of the Year. Ladies and gentlemen, Dr. Njar.
I would like to start by thanking the president, of UMB, Bruce Gerald, for his kind his introduction. And then, of course, I don't have a choice. I will thank my dean, Dean Reese, who gave me that flattering, this introduction. So Dean Reese, I will not disappoint you. Okay, so good afternoon, everybody. And I'm pleased, to be here with you today in person and it's virtually, for you to listen to my Entrepreneur of the Year, this presentation. I am, very humbled and honored, and I'm grateful, to be chosen for this award. I thank Bruce Gerald and the committee that found it, this worthy, to select me, for this award. As you would hear, this throughout, my talk. The credit of this award goes to, my former students, the present students, and, the postdoctoral, these fellows, and my longtime, colleague, Dr. Angela Brody. You would see that throughout this talk, they all helped, to drive our research over, these 2 decades. I like, always to use my pointer. So I would actually stand, on the side and advance, the slides and talk. This is the title of my talk, discussing, these small molecules. There's a subtitle there. That's drug discovery. This ingenuity, and serendipity, or is it the other way around? And as you can see, for the most part, for what I'm going to present today, you would ask that same question. My talk will be divided into 2 parts. In the first part, I will talk about prostate cancer, this innovation. In there, I'll discuss that's galantaron, the development of galantaron, and the next generation is galantaron analogs. In the second part, I will talk about breast cancer pharmaceutical innovations. And there I will discuss that's our Ramba molecules and the MNK, the truncating agents. And I will, also, this highlights, that's isoprene pharmaceuticals. I'm required, by UMB, that each time I give a talk, I have to make, these disclosures. That's part of, that's the management plan that I have, the conflict of interest. So in the first part, of course, this technology was discovered here at UMB. I lead, the group, in collaboration with Dr. Brody for the galantaron, this technology, that was licensed, to Tokai Pharmaceuticals, but it's now licensed to ESL. I'll discuss that, this later on. And this, was the signing ceremony, for the licensing to ESL. We all know, that's our former president, who is now the chancellor of University of Maryland system. And then I am also the lead, this inventor of the Rambas technology. It's the president, Bruce Gerald, and Dean Rees. Drug development, from academia, I could be called academic, this research. But some drug discovery efforts, are anything but academic. Okay, research means this careful, this diligent search. Studios, this inquiry of examination. I mean, very noble and kind words. Okay, but academic is a horse of a different color. Academic means, in its noun form, it means it's a person who is very learned but inexperienced in practical matters. Adjectival form means having no practical or useful significance. It's a synonym, with pen. Okay, pedantic. Academic, that means it doesn't matter. President Gerald, I want to do things that matter. I certainly don't want to be associated with the term. It's pedantic. Okay, this is an excerpt from the Dean's letter, from Dean Reese's letter to me, congratulating me for this award. And in one of the sentences which I picked up, I quote, "We need some more research, educator, these entrepreneurs like you." I already told you what academic means. Okay, this entrepreneur also is very kind and is noble. It means one who can organize an assumed risk of a business. It's derived from the French word, this entrepreneur. That's to undertake. Entrepreneur, and the synonyms creativity, and all the good things. So if I ask a question now, can the person be an academic and an entrepreneur? We don't have time to discuss that. So I would say my answer to all of us, and I believe you would agree with me, is yes, yes, and yes indeed. So I would this love for a day when somebody's called an academic, it should mean that does matter. So, that tax goes on to the leaders of this university. I mean, they should actually make a case to the Oxford Dictionary personnel. All right, ladies and gentlemen, I'll now go on to tell you, that's the story. We started out trying to identify a specific CYP17 inhibitor, but we ended up with a multi-target, this molecule. And that is under prostate cancer. These androgens, would stimulate the growth of prostate cancer. And it was shown that if you inhibit the synthesis of these androgens, you bring down the levels and the tumors will regress. So the main sources actually of androgens in humans or in males is from the testes and adrenal glands. And of recent, we now know that the prostate tumor itself is able to synthesize its own androgens. We should be familiar with the male reproductive tract. The urethra, that's the prostate, it sits on the, that's the bladder, and it surrounds, that's the urethra. That's Charles Higgins, won the Nobel Prize, in medicine in 1996 for the discovery of castration for the treatment of prostate cancer. So what that means is that all he did, was to remove, that's the testis. But because we wanted something this much better, we believe that if you inhibit the synthesis of the CYP17, you can block the production from all sides and not just from the testis. I mean, those are the stages actually of the disease. Okay, but what kills the patient? Okay, that's metastasis, and it goes all over the body, mainly to the bones and the lungs. So, and it's known that okay, the 5-year survival rate, I mean, for metastatic disease is very low, 28%. If you compare that to 100% for when the disease is not metastatic. So indeed, it's important that we try to develop new drugs. So this slide here shows there's a biosynthesis of these androgens, from cholesterol. And the enzyme CYP17 is a two-step this reaction, a hydroxylase and a lyase. So what happens when you inhibit that? This is what you see. You get a down regulation actually of the androgens. Okay, that's irrespective actually of the source. The action of these androgens, is illustrated here in this slide. Okay, that's testosterone gets converted to a more potent androgen DHT, shown here. And then it interacts with the AR and binds to the AR. Okay, there's dimerization and phosphorylation. It enters to the nucleus and binds to the androgen response element to initiate that proliferation of the cancer. So our design strategy was based, on the knowledge of the enzyme, the architecture, and the mechanism also of how the CYP17 works. Okay, the crystal structure of the enzyme was not known at the time. What is known now, but has not made that much a difference in terms of the design of these molecules. And the whole idea was, this is the substrate immediate of the enzyme. And if you modify it around this region here, the C16 to C20, to have groupings, we can disrupt its activity. You should be able to inhibit the enzymes. That's shown in this model. Okay, critical to this, to galateron discovery was we invented, that's a new reaction, where I would spend some time to explain this. The whole idea was we wanted, to hydrolyze 6 to 7 with hydroxy, but we didn't get this. Instead, there was a hydrolysis and we got the substitution. We reasoned that under this appropriate conditions, you should be able to form azoles. And we made a whole family of these new compounds. And that allowed us to synthesize a lot of CYP17 inhibitors. At the time, this was very unique. In the literature, we didn't know any steroidal compound in which the azole is linked to the steroid nucleus via nitrogen. And of course, this reactions, this led to that's galateron. So we can ask the question, ingenuity and serendipity, or is it the other way around? Okay, the synthesis is shown here, which I'm not going to go through. But the important thing was that our synthesis was that this adopted in the clinical development of galateron. In this slide, I would show you the impact of that galateron on different models, of prostate cancer. In the first model, they depend on the AR, and we compared it with castration, and one of our lead compounds, we compared it with Casodex. That's an approved drug. And in this study, we did something unique. That's prevention. In all these studies, you allow the tumors to grow, to a certain size, and you start treating them. But here, a day after the cells, the prostate cancer cells were inoculated, we started treatment, and you can see for, 93 days, I mean, the tumors did not grow at all. And then we now compared, with that abiraterone shown here. The CRPC, which is metastatic, again, we were able to show that our molecules are quite effective. So in essence, we have shown from all our studies over the years that galanterone is more efficacious than castration, Cosidex, abiraterone, or enzalutamide. So in trying to summarize, what we now know about this galanterone, and this figure, was generated by one of my graduate students about 10 years ago, and I'm still using it. So what it shows is that this galanterone is able to inhibit CYP17, it's able to block the action of androgens on the AR, and it's also able to cause a downregulation. And some prostate cancers do not express the androgen receptor, and he was able to show, that's Rob Bruno, that these molecules would induce this ER stress response. And he also noted that there was an upregulation for ERF42E. That then gave us an indication that our molecules could inhibit this oncogenic protein translation shown there, and we have evidence for that. So we did these SAR studies, and the highlight here was that the molecules do not have to bind to the ligand binding domain of the AR to be active. I would skip a number of slides here. So this is another phase 2 study for these molecules, where they to try out this design. And again, if you look at that's the PSA, these levels, it was it's impressive. It's based on the studies, the design of the phase 3 was done. So Tokai went back to their phase 2 data, and because we had shown that galanterone could inhibit the splice variants, they found out that 6 out of the 7 patients that had splice variants, that they responded to the therapy. So their phase 3 clinical trial, was based on, just trying to see the impact in patients in the clinic. However, this clinical trial failed, but before the clinical trials, there was a publication by this group which says clearly that because of their trial design, it was guaranteed to fail, and that the FDA had even warned them of the trial design. And look at their phase 3 study. If you compare it to the other drugs, the sample size was quite a problem. So based on what we know, I think that the molecule fails in phase 3 because of the trial, this design. Another thing too that happened here was that the insiders in the company, they were all selling their stocks during the planning of the phase 3 clinical trials. So, to rescue this molecule, that's ESL has, now, just license it. And then, then based on, some studies in my lab, I must point out that David Weber was instrumental for us to get that galanterone from that Tokai. He actually paid for it for the storage. So ESL, is now developing this. I would do one thing since I don't have much time. The clinical trials, it's in Jamaica. So I'll tell this because Dean Reese is here. So the reason, I'll show this. So the reason why the clinical trials are in Jamaica is that, the founder of the company, the founder of ESL and ETL is from Jamaica, But another issue is that, okay, the Caribbeans, okay, has the highest, okay, mortality rate for prostate cancer. So if you look at this, if you compare the countries, the Caribbean has the highest. So, I mean, we're also developing this compound, I mean, pancreatic cancer. I'll skip a few slides here and just show you what we're doing. Okay, that's Kevin Cullen, of course, is paying for a phase 2 clinical trials in pancreatic cancer here at our cancer center. It's based on results, I mean, from my lab. We'll skip some slides. These are the current projects that we have. Of course, we need to have IPs for protection, and that, of course, Jim Hughes's office is very helpful in doing all this. These are the students, postdocs, and these are the ones out here. I will just keep, how much time do I have? Okay. I'll just show this slide and then skip a couple of slides. Okay, ATRA, all transrotinoic acid. I mean, we derive that, I mean, from our foods. And it was shown to be effective in APL. It's now also been approved, okay, that's for neuroblastoma. I'll just skip a couple of slides. Okay, but this clinical failure is because, of course, there's an inhibition. I mean, sorry, this ATRA gets converted to the hydroxy. So we now decided to see if we could inhibit the CYP enzyme. And if we do that, of course, and we now got, okay, quite some potent these inhibitors. And the design strategy for ATRA here, I mean, was quite similar to what we had for the CYP17. This is the synthetic scheme for that. But we were now able to show, okay, that our lead molecule does degrade the MNK, okay, this 1 and 2. Remember again, we started out with inhibiting CYP26, and we now find these other molecules. This is an interesting slide here. We all know that in most diseases, the AKTPI3 pathway, the MAC-RES pathway, P38, they all converge here in a protein translation. And this is found in most diseases, not just, I mean, in cancer, in Alzheimer's, whatever. So we have shown that our molecules are able to inhibit this. I will just skip quite a number of slides and go on. So it's also been shown that the molecular target we're looking at, the MNK, is involved, okay, that's in most cancer, breast cancers, and triple negative breast cancer. These are cell lines which express this. So we had our first, okay, this publication on this, where we showed that we published that we discovered the first MNK regulating agents which block EIF4E and induce apoptosis in cancer cells. And we have shown this also in prostate cancer. Okay, this is the mechanism. There's degradation of MNK, and then you have a degradation of phospho-EIF4E. So we now have done this in vivo studies where you can see when we dose this molecule to this compound, this is the control. You can see the huge tumor, while the other groups actually treated with our compounds were very effective. Okay, the body weights were fine, which shows that there was no toxicity. There was strong target engagement, as you can see here, MNK and MNK2, phospho-EI4E. That's the quantification, that's the apoptosis. And according to the NCI criteria, they consider that our molecules are highly effective. And we have shown this in other these models. We also did some studies in tumors, okay, derived, okay, that's from patients from our hospital. And we collaborated, okay, we ran slapides. And our lead compound, you can see, is quite effective compared to the control. And the targets we're looking for, MNK1 and 2, are all down. We also just repeated this experiment. I will skip these experiments here. So when we now had all this impressive data, we had a discussion with the FDA group, and they told us, okay, since you have now formed this company, Isoprene Pharmaceuticals, instead of going to apply for a phase 1 SBIR grant, you can apply for this direct to phase 2 SBIR. We partnered, of course, with, these are the students and postdocs who worked in this. We founded, Isoprene Pharmaceuticals with the help, of Phil's group, Rana, Jim Hughes, of course, is the head of everything. But I worked closely with Rana, with Phil, with Nancy, with Sue, and all that. So they all helped us. We then wrote the grants. In writing a phase 2 grant for SBIR, one of the critical things, in addition to the data you have to have a plan on how to move forward, commercialization plan. And they did all that for us. These are, the officials. That's Rana. That's Vidya. It's a former postdoc of mine. So she is the chief scientific officer of the company. And that Sue is the one who does the financials. You might want to wonder and ask Bruce Gerald and Dean Rees, Vincent Njar is still a professor in the School of Medicine. So how come he's an official of Isoprene? Well, I have what you call a UMB approved this conflict of interest management plan, which means that you tell them this is what you are doing and it's approved. So you can still be here on campus as professor and still found a company. All right, so we then applied for this direct to phase 2. We're just awarded this 2-year direct to phase 2 grant. And the idea is to move things towards phase 1 clinical trials. We're partnering with the company Davos, these pharmaceuticals, which is involved in development of molecules. These are the specific aims actually of the phase 2 there, and we hope to finish this within 2 years. So these are people who have funded our research. And here I would like to just acknowledge that's the champions of my UI award. Of course, Dean Rees, the nominator, the external references, that's Doug Covey and Doris Benbrook. The internal, these champions, of course, I would acknowledge them. My chair is here, Peg McCarthy. Kevin Cullen is not here, director of the Cancer Center. And of course, David Weber, the director of CBT, and the interim, this co-director of IBBR is also here. And then, of course, the awarder of the Entrepreneur of the Year is Dr. Bruce Gerald, UMB president. For those of you who don't know, he also has another title. He's a professor of surgery in the School of Medicine. And last but not the least, I'd like to thank my other halves. Rosalyn is here, my wife, and my son, Vincent, he's also here. I'd like to thank all of you. I'd like to thank, the committee, Bruce Gerald, and all attendees. Thank you.
So we are now going to get into the Q&A portion of this presentation. So if anybody has a question, you can raise your hand. Just wait for the mic to get to you, so that way we can hear your questions. And for all of our Zoom listeners, go ahead and type your questions into the chat and I'll relay them to Dr. Njar.
Vincent, did I hear you say that the galadron molecule was also impacting the EIF?
Yes,
or F,
yeah.
so that you could actually have sort of a multiplexed approach to
Yeah.
inhibiting that?
Yeah.
Is that right? Yeah.
That's right.
That's fascinating.
Yeah.
So Vincent, you probably know what I'm going to ask, but the inhibition of translation initiation is so powerful, and the relationship of that to pancreatic cancer. Could you just make a comment about some of the things we saw in the cancer center with KRAS?
Oh, okay. So, all right. So that's fine. I guess, I mean, I had a slide on that, but because of the time constraints, I think I have to move forward. So let me show you. Getting there. Oh, come on. Yeah, I think, no. Okay, so actually, let's go back. Okay, so pancreatic cancer, I mean, there's this mutation of the KRAS in a lot of the cancers, and it's a key driver actually of the disease. And it does impact actually, I mean, on protein translation. So if you look at this pathway, I mean, RAS is right up there. So it does impact on that. And it's been shown, okay, clinically, that upregulation of the phospho-EIF4E, that's this here, okay, that correlates with the grade of pancreatic cancer grade onset, okay, and was, okay, this prognosis. I mean, so because we have shown, okay, that our molecules are able to impact on that, that's how, I mean, we now started developing these inhibitors for pancreatic cancer. So if you look here, we conceived the idea in 2014. I was able to apply for an R21 grant, which was successful. And then Tokai was also interested in the development. So there was, okay, the sponsored research, and we had our first publication actually, which shows, if you read the title, galatron and its analogs inhibit the MNKF4, it synergize with gemcitabine. That's the lead drug for this disease, as we know, and impede cell migration, invasion, proliferation, inhibition of tumors. So that's how, I mean, we went on, I mean, to the clinical trial story. Okay, the clinical trial story was possible because we had these tablets. So we should give thanks, okay, to David. I mean, I didn't have the funds to pay for the tablets to be stored. Okay, but the other thing also you should think about, okay, these tablets cost, okay, that's a lot of money, about $3 million. And they were actually going to throw them away. I requested for them. I asked them the question, okay, so what are you going to do with the galatron that you still have? They said they're going to toss it away. So I asked, I mean, can you transfer it to UMB? And that's what they did. Okay. Okay, then Kevin Kulan, of course, I mean, when he saw our data, I mean, he now, was quite impressed, and decided to fund a phase 2 clinical trials, which is ongoing as we speak.
Vincent, first of all, congratulations on
Thank
an
you.
excellent tour de force of your work. Now, what is the unifying hypothesis that galadrone would work against prostate, pancreatic, and a third cancer, whichever?
Okay, so I mean, I really believe it's because of the protein translational target. So when we even started thinking of that's galadrone in pancreatic cancer, there's some phase 2 clinical trials which shows that the AR, the androgen receptor, is also upregulated in some pancreatic cancers. So that's what I actually started thinking about first. But eventually, we now found out the impact of that some protein translational complex. And that's why, we now wrote an R21, like I said. The R21 grant, as you know, is supposed to be a high risk, it's high reward grant. We got it. So the impact actually, I don't have the slide, but if you look at the effects that we saw, that's galadrone, and the next generation compounds, which are even better, about, that's about one-fold. One of the issues we had, that's with galadrone, was the dose. Remember I told you that they're using in the clinic. And that, has to do with the pharmacokinetics. But the newer molecules that we have now, the next generation compounds, which I didn't have time to let's talk about, they are much, much better than galadrone in terms of, and we did a study which showed that they are much better because of their impact on the MNK, the protein translational pathway. So the protein translational pathway, in fact, is a pathway that if we're able to develop these compounds, it can work for almost any disease. I have a slide here. Let's see. Okay. How do I get this off? Do you have a minute? It would be easier. How do I get this off? Okay. All right. Okay. All right, so I think I have a few slides we can discuss this now. So again, like I said, the PI3K RAS, this pathway, they're majorly upregulated in most diseases. It's not just cancers. And they all impact here with some protein translational complex. And then this, of course, goes on to make these oncogenes. So what happens is that, that's the PI3-AKT pathway. When it's dysfunctional, the binding protein, will release the EIF4E, which binds with the EIF4G, to form this complex. But the problem, for this to be, this oncogenic or involved in any disease, this, has to be phosphorylated by this MNK1 and 2. And what we found out, with our molecules is they induce the degradation of MNK1 and 2, proteasomal degradation. And if you do that, you prevent this phosphorylation. And once you prevent this phosphorylation, of course, you inhibit that complex. There's a question that I get asked a lot, which so I had this slide, but I couldn't explain it. So you might ask, you are inhibiting, that's protein translation. That's all of us, of course, we need, that's our proteins, to be translated. They have to be translated. You go on from the DNA, which makes your mRNA. The mRNA, of course, goes on to make the proteins, right? Okay, so if you ask this question, why won't inhibition of ERF4E kill all cells and not just the cancer cells? And the answer is very simple. In our bodies, we have two main types of these mRNAs. You have what you call these mRNAs, and are those which are used to make the normal proteins that we need, the beta-actin, GCAD. And then you have what you call the weak mRNAs, and those are the ones involved in formation, in this translating to oncogenes. This here would be formed even, when, the levels of ERF4E are limiting. You would form this very well. However, these other ones, you need an upregulation actually of ERF4E for them to be formed. And these are involved. And when you inhibit, of course, this, that's what happens. Which means that if you reduce the ERF4E activity, it would have a minimal effect on the total protein synthesis and therefore minimal effect on the normal cells. The next slide, I think it's the one that, when I discuss with some of my colleagues who ask this question, they appreciate it. So if you look at this slide here and look at, that's protein synthesis, the rate, and look at, the normal growth, and that going on, the tumorigenesis, and to metastasis. Here, at this level, where the ERF4E activity is very low, you can see these housekeeping genes are formed, to the maximum. But in most diseases, there's an upregulation actually of this pathway. So you can see the binding protein here, that's phosphorylated. When it's phosphorylated, it will now release the ERF4E. So you have an overexpression of that. And that goes to bind to the ERF4G, which forms the complex. And for this to be, that's oncogenic or to be involved in any diseases, this phosphorylation is important. And it's phosphorylated by the MNK, this 1 and 2. So in fact, it has been shown that, that's in vivo. Those are the only two known kinases for ERF4E in vivo, the MNK1 and 2. And our molecules are able to induce the degradation of those. So in essence, all you are doing actually in the disease state, you are inhibiting the activity of ERF4E, which is in this complex. Why do I've been talking a lot and mentioning ERF4E. You have, these components that form this complex. Of all of these complexes which form the ERF4F complex, the ERF4E is what we call the most limiting. So that's why if you inhibit it, it's very impactful on the ERF4F, this complex. I wrote a review article actually on this, I think in 2017, how to target, protein translation for this. But if you look at, the literature in Alzheimer's, in a lot of diseases, the AKT, that's PI3, use pathway and the RAS are all upregulated. And it has been shown also that the MNK, protein translational pathway, are upregulated.
That's all the time that we have left for questions. Thank you so much, Dr. Njar, and congratulations. Everyone, let's give him another round of applause. Congratulations. So now we're going to be moving on to announcing our 2021 faculty patent winners. And to do that, I would like to welcome the Associate Vice President for the Office of Technology Transfer, Phil Ravalato.
Great. All right, well, thanks everybody, and thank you, Vincent, for an excellent presentation.
Thank you.
And thank you very much for inviting so many members of our team here today. 'Cause with limited attendance, we really appreciate the invitation. Anyway, I guess we're running a little late, so I'm going to start off by just saying, you know, we added this segment just a couple of years ago to honor our inventors during the Entrepreneur of the Year ceremony, because getting a U.S. patent in this day and age is a very significant accomplishment. It's a very difficult thing to do, and I think it's a nice thing to really honor all of the UMB inventors that have had U.S. patents issued during the most recent year. Before I read off on the list, I just want to remind everybody who tech transfer is and how we're sort of related to this process. So just very quickly, we are 3 teams. Okay, there's a licensing team, which I think most people are pretty familiar with, an intellectual property team, and then we have a new ventures group, which Vincent alluded to during his talk. So these are the folks that work with the USPTO. They help navigate the compliance issues with the federal government, other funding agencies, and they help to commercialize the technology by finding good partners to license the technology to. You know, so these first 2 groups, the IP team and the licensing team, will do probably 130 to 150 new disclosures every year. There'll be 200 to 250 to more new filings every year. We will do probably 70 term sheets with companies. We'll execute 50 new deals every year. And we start 8 to 10 companies. Last year, we actually started 11 despite COVID. So I want to thank everybody in those groups for all of their hard work and efforts and all they do to help move this IP through its processes. And very quickly, I also want to acknowledge our new ventures team, which we have Rana here, she's the head of. And this group is a newer team. You may be less familiar with them, but since their inception, they act as an accelerator. They've helped accelerate 24 UMB technologies. They've spearheaded 16 investments in 11 UMB startup companies, and they have launched 3 companies themselves, the first 2 of which were sold successfully. And the most recent one, which is Dr. Njar's, and his technology is actually the main asset in Isoprene. And they just received a direct to phase 2 SBIR for $2 million, which is an incredible accomplishment. So congratulations to them, congratulations to Isoprene. And with that, I'm going to go through our list of new US patents and inventors for the past year. I have a slide. There's contact information. In years past, we have handed out the plaques at this presentation. Obviously, we can't do it this year. But if you would like to set up a time or stop by to pick up your plaque, there are folks in the office every day if you want to do that. Or you can reach out to Gail Knott. Thank you, Gail. And she will help make arrangements for you to be able to get your plaque. So I'm going to go through the list, which I think you can download off the web or the email. And it's in alphabetical order by school, starting with the School of Dentistry. And so the first awardee, the first lead inventor is Hanpin Fang for tetraspecific octameric binding agents and antibodies against C. diff, toxin A and B for the treatment of C. diff infection. This technology is actually part of Dr. Fang's startup Fzada and is doing very well. Second patent is Radi Masri, our second inventor, for compositions and delivery methods for treating dental infections, inflammation, sensitivity, and for use in dental restorations. This technology has received an MII from TEDCO, and Dr. Masri is working very closely with our new ventures group on that. Next up is Hawken Jew, and he has a co-inventor, Michael Weir, for rechargeable calcium phosphate containing dental materials. Dr. Jew is a very prolific inventor and has spoken at the Hopkins UMB Alliance meeting several times. and his technologies have garnered a lot of commercial interest over the years. And then last for the School of Dentistry is Rana Yunus for a method for monitoring cancer and for regulation of semaphore and 4D to improve cancer immunotherapy regimes. Dr. Yunus also won an MII from TEDCO for this technology. And so those are our winners, or sorry, our awardees from the School of Dentistry for the past year. Next is the School of Medicine, and I think the first one on everybody's list should be Tony Antalis. And her patent is engineered anthrax protective antigens proteins for cancer therapy. This is also a very intriguing technology. It's received quite a bit of interest from commercial partners over the past few years. Next is Joseph Barton for a method for assessing fall risk based on tracking data of a moving target. This technology is currently being moved forward at the prototype stage, but has already attracted a couple of commercial inquiries. Steve Bernstein is next. He has 3 UMBC co-inventors for screen printing tissue models. This is co-owned with UMBC and is actually the subject of one of their startups, which was launched just last year. Kurt Sibin actually is the next 3 patents on the list. They are all co-owned with Inventors for Princeton or GPB Scientific. Methods for preparing therapeutically active cells using microfluidics. Methods and devices for high throughput purification. Methods for preparing therapeutically active cells using microfluidics. And again, all 3 of these technologies are co-owned or have been licensed to Princeton startup GPB Scientific and are all doing well. Next on the list is Eric DeLue, who is actually no longer at UMB, but he has 2 patents, and he's got 3 co-inventors who are still here. Steve Fletcher, Alex McCarroll, who's with us, and Jamal Shawwan. And both of those patents are entitled small molecule lipid 2 inhibitors. And these technologies receive funding from the Life Science IP Fund. And I know Dr. DeLue is interested in starting a company around them. Next is Matt Freeman for multivalent vaccines for rabies virus and for coronaviruses. Dr. Freeman is another very prolific inventor here at UMB. And many of his technologies have been licensed, subject to sponsored research, or just in general received a lot of commercial interest, particularly over the past 18 months, as you can imagine. Next to actually is Todd Gould. He has, first one is co-owned with Hopkins, DNA methylation biomarkers of postpartum depression risk. And the second one is crystal forms and methods of synthesis of 2R6R and 2S6S hydroxynorketamine, co-owned with NIH. And this is actually the subject of a phase 1 clinical trial that the NIH is sponsoring. The next 2 are Elliot Hong for system apparatus and method for transient electric field detection and display, and systems for controlling magnetic fields and magnetic field induced current. These technologies are part of a very nice ongoing collaboration with UMBC. Thomas Horniak is next with co-inventor Sandeep Joshi for functional methylation of neurons with melanocyte stem cells. This was one of several technologies that was pitched at Bio recently. Next is Peter Hugh with co-inventors Shimeng Yang and Colin McKenzie for method and apparatus for predicting a use for blood transfusion. This is another terrific technology. The inventors have been awarded several years of DoD funding for the clinical evaluation of this technology, and they're actually currently pursuing FDA clearance. And then the next 2 on the list, Dr. Anthony Kim, and I moved up Dr. Graham Woodworth because they're part of a very, very inventive team. So Dr. Kim with Dr. Woodworth and Dr. Winkles decreased that adhesivity receptor targeted nanoparticles for FN14 positive tumors. And it goes hand in hand with the Dr. Woodworth lead technology, which is targeted structure-specific particulate delivery systems. Again, this very promising technologies, very prolific inventor group. They've recently been awarded a TEDCO Phase 1 MII for this. Next is James Lyons. He is no longer at UMB, but his co-inventors Joseph Staines and Christopher Ward are. For multifunctional fluid flow device, this was successfully optioned to a medical device developer recently. May Montasser. This is co-owned with Regeneron, and of course, this technology is part of a very successful collaboration with that company. Sarah Murthy is next on the list for system and method for assessment for cardiac stroke volume and volume responsiveness. Dr. Murthy is another prolific clinician and inventor, and she is a successful entrepreneur with her own startup, Looking Glass Technologies. Vincent Njar is on the list. I actually have your plaques with me, so you can get them today. And I have two from last year for you too. And again, this is for 13-cis ramborotanamides that degrade MINCs for treating cancer. And you know, we just heard from Dr. Njar. This technology is part of the highly successful NBI startup, which, as I mentioned, was just awarded a direct to phase 2 SBIR. Next is Marzina Pazgir with co-inventors George Lewis and William Tolbert. Stable immunogen-based interdomain of HIV-1 GP120 for inducing immunity against HIV. And this technology has been under recent evaluation by one of our most senior EIRs. So we're very hopeful there. Next is Jeremy Polf and techniques for producing an image of radioactive emissions using a Compton camera and Compton lines. This clinical device prototype is currently being tested, and the invention, as you can imagine, has tested quite a bit of commercial interest. The next two are Jacques Revell with his co-inventors Michael France, Lindsay Rutt, and Bing Ma. 2 patents, both entitled Microbiome-Based Informed Method to Formulate Live Biotherapeutics. And both of these technologies are actually part of very successful UMB startup, Luca Biologics. Next, we have Charles Sanser with co-inventor Steve Ludwig for interbody cage with spill-free biological material compartment. And this technology is moving forward very nicely. We actually now have a prototype for it. Next, we have Dr. Raj Sarkar for device and method for automated emergency arterial sheath placement. And Dr. Sarkar is working very closely with our team, with UM Ventures and with SiteMiners looking to apply for an MII soon. Next up is Amit Sawant for deformable lung model apparatus. This actually is a very novel phantom for motion management quality assurance in radiation technologies. The next two are Mark Simard with co-inventors from Remedy, and this is for methods for reducing or preventing intimal damage caused by mechanical stimulation of endothelial cells. And the second one is targeting NCAA ATP channels for organ protection following ischemic episodes. And Dr. Simard is, of course, another very prolific inventor, a former Entrepreneur of the Year himself, and many of his technologies have become the basis for Remedy, which is one of our startups, and are now part of Biogen, undergoing phase 3 clinical trials. Next is Sharon Kenneth and co-inventors Mike Levine and Rafi Simon, who is no longer at UMB. And that is for compositions and methods for producing bacterial conjugate vaccines. And this technology is licensed to Barton Biotech. Next, we have Gerardo Vasta with co-inventor Elijah Wang at College Park for inhibition of galactin-3 binding to airway epithelial surface to treat or prevent septic shock resulting from influenza and subsequent pneumococcal pneumonia infection. And this technology, Dr. Vasta, was also awarded a phase 1 MII from TEPCO. Next is Stephanie Vogel with co-inventor Katarina Richard for compositions and vaccines comprising vesicles and methods of using the same. Dr. Vogel is an exceptional inventor, many technologies disclosed over the years, and she has been a long-term and very valuable member of the scientific review committee. The next two are Elijah Wang, who is now at College Park, along with his co-inventor Wei Huang. These chemoenzymatic glycoengineering of antibodies and Fc fragments and core fucosylated glycopeptides and glycoproteins and uses thereof. Dr. Wang is the co-founder of Glyco T, which these technologies are a part of, and it's a UMB startup doing extremely well. They recently announced a successful sublicense with a major Japanese partner. The next two are Dr. Jeffrey Wolf, with the first one is with co-inventor Amal Isaiah for automatic ultrasound titration of continuous positive airway pressure treatment for sleep apnea, and system and methods for emergency apneic oxygenation. Dr. Wolf is another extremely talented, active clinician inventor. He's part of MDC, and this ultrasound technology has been successfully licensed to UMB startup Tesserae. We talked about Dr. Woodworth's technology earlier. So the next one on the list is actually John Wu with self-expanding cannula. And this, of course, is licensed to highly successful UMB startup Breathe. Co-inventor Dr. Bart Griffith was the co-founder of Breathe, and that company, of course, was acquired by Abiomed just about 18 months ago. Next is Howard Zhang for system and method for optimizing a treatment plan for radiation therapy. His co-inventors include Warren D'Souza and Byung Young Yi. And this actually is another great team of researchers that have been involved with a lot of very successful projects and sponsored research and commercial agreements over the past several years. So that is the School of Medicine. And in the last school we're going to cover is School of Pharmacy. And we have four inventors there to list, four new inventions. First is Stephen Fletcher, and for small molecule inhibitors of the MCL1 oncoprotein and uses thereof. This technology has also been the subject of sponsored research interest. Vijay Avaturi with co-inventor Joga Gabora for a method of administering sotalol IV switch. This was successfully licensed to Altathera Therapeutics, which recently achieved its first commercial sale. Next, we have Fengjin Zhu with co-inventor Yan Shu for Wnt signaling pathway inhibitors for treatment of disease. And again, this is another UMB Phase 1 MII award winner. I believe Dr. Zhu actually has won 2 of these awards, and they're not easy to get. So congratulations on that as well. And then last, but certainly not least, we have Bruce Yu and Mark Taraban for biopharmaceutical aggregation assessment and counterfeit detection using magnetic resonance relaxometry. This is another technology which has received a lot of interest over the years. It's being developed through National Institute for Innovation in Manufacturing Biopharmaceuticals, which we call NIMBLE, and is also won a UMB NII from TEDCO. So that is it. A lot of very exciting technologies across a wide variety of fields, many of which have been licensed or commercialized or gone into our own startups. So again, I want to say congratulations to all of these inventors who have been issued a patent, a US patent in the last year, and all of their co-inventors. Thanks for all that you do, and congratulations.
Thank you so much, Phil. Congratulations to all of our patent awardees, and of course, congratulations to our Entrepreneur of the Year, Dr. Njar. And thank you all so much for attending today's event, both in person and virtually. Hope to see all of you back tomorrow for our Researcher of the Year event. That's at 4 o'clock. Same room, not the same Zoom, but on Zoom and in person as well. So thank you all so much. Have a wonderful evening.
Thank you.